dear ladies and gentlemen, dear Alpha friends. It's my pleasure to welcome you today to this presentation of two high-level medical research projects that are financed by the European Commission. We call them Layman events because we try to get the high-level medical data and treatments and research and put them into the words that would be easy understandable to general public and also to patient groups. So sharing medical research data with general public, patient organizations as we are, and also patients is of paramount importance for several reasons. Firstly, transparency in research findings fosters trust and accountability in the healthcare system, allowing us to make informed decisions about our treatment. Furthermore, disseminating data empowers patients with knowledge, enabling us to actively participate in our healthcare journey and advocate also for our needs. Patient organizations as ours also benefits from the access to research data as it equips us with evidence-based insights to better support our members and also to advocate for policy changes at the highest levels in the European Union. Additionally, sharing research data can also lead to accelerated discoveries as it encounters collaboration among scientists, healthcare professionals, public, and also potential anyone who is interested in the topic. This can also advance medical breakthroughs and improve healthcare outcomes for all. In essence, the open sharing of medical research data promotes transparency, patient empowerment, collaboration, and ultimately the advancement of healthcare for the benefits of the society as a whole. I leave you today for sharing the event into the hands of Ms. Veronika Przetichkova, who is the leader of all European affairs at European Liver Patient Association. Ms. Przetichkova, the floor is yours.
Thank you very much, Marco, for a kind introduction. Without further ado, I will give the floor to our speakers. So first of all, welcome everyone who is watching us. This is the very first Layman event of the European project Genial, understanding gene-environment interaction in alcohol-related hepatocellular carcinoma. With us today, we have amazing speakers, Mr. Mathieu Dionin, Professor Eric Trepot, Dr. Patricia de la Cruz Ojeda, Dr. Tine Tricot, and Professor Pierre Nahon, who will introduce the project, the main objective, what are the aims of the project, but they will also touch on the objective and work of a specific work package inside of the project. So without further ado, the floor is yours.
Yeah, wait, normally you can see the slides, right?
Yes, we can see the
Okay,
slides.
fantastic. So I will try to stay on schedule. So I'm very happy as the coordinator to present this project that is called Genial. So Genial stands for Gene-Environment Interaction in Alcohol-Related Hepatocellular Carcinoma. So you basically understand that I'm going to talk to you about or present you a project that is about genetics, so the genes and the environment. And the main focus of our project is related to liver cancer and liver cancer that arise on the background of alcohol, which is a major problem, as we can see on the slide, in Europe. So looking at alcohol's impact on health in Europe, you can see that in 2019, 6.2% of all deaths in Europe were linked to alcohol consumption. And it is really the biggest health issue caused by alcohol, where you can see on this graph, where in fact cancer, and followed by liver cirrhosis. And among the alcohol-related cancer, liver cancer ranked just second. It is then therefore cancer. And alcohol-related liver disease is extremely prevalent yet overlooked. It is the leading cause of liver cancer, cirrhosis, and liver-related death in Europe. And what you can see here, it affects life. It mostly impacts working adults. So adults that are, around their 40s or early 50s. And there is really a need of research, because you can see that 30% of all liver disease deaths are in fact, alcohol-related. But unfortunately, despite its impact, there's only 5% of liver disease clinical trials and publication that focus on alcohol-related liver disease. And this is where we hope Genial is going to fill the gap. So Genial aims to address this critical lack of understanding and research on ALD, so alcohol-related liver disease, especially in relation to liver cancer. The project is ambitious, and it aims, as I said, to a better understanding, treatment, and management of ALD. So the slide shows basically an overview of the structure of the project. It's a collaborative effort. You can see here that there are 5 work packages that are interconnected. And well, I will not go into the detail because this is going to be the focus of the work package leaders. But you can see here that in work package 1, we are focusing on identifying, let's say, genetic variants in large cohort, retrospective cohorts. Work package 2, we're going to try to understand how these genes interact in terms of environment and environment more, let's say, at the basic science level. Then in work package 3, it will be something at the population level. Work package 4 is, in fact, related to communication and is led by ELPA. I will say a word to that in a minute. And work package 5 is basically the management. So it is a very busy slide, but I would like to emphasize that it's a pan-European effort, and that highlights that many institutions in Europe are involved. And it showcases the diversity and the strength of this collaboration. And let me say a word about the role of ELPA in this project. So ELPA is really a central involvement. It will be actively involved in all work packages, as you can see here. And ELPA is also leading the dissemination, spreading awareness, knowledge, and training related to this project. So the role of ELPA is really critical because liver cancer predominantly affects individuals with chronic liver diseases. And by having a patient association like ELPA at the forefront, it ensures that patient perspective is central in all this project. That's exactly what we aim for. And let me say a word about the general impact. So this scheme is really what are, you know, the overall aim of this project. So of course it is understanding. It is in the understanding cluster. So we are going to try to understand how gene and environment, you know, interact and promote liver cancer. But we also would like to go further and have a role in prevention by identifying all the environmental factors that promote alcohol-related liver cancer. Also a role in early detection, and I'm sure that Pierre in work package will say a word about it. We would like to facilitate early detection that is crucial for improving survival rate and contribute to stratify the risk of patients using advanced tools such as artificial intelligence. And although it's not the main objective of this project, we hope that we may have an impact in refining screening strategy and potentially identify candidate drug targets. So very quickly, you can see that's a 5-year project. We've been very well funded by the EU, and we are very, very happy. And this is the beautiful team, and we really hope that we will deliver something impactful and that, you know, patients will not be forgotten because they are really at the center of this project. Thank you very much.
Thank you very much, Eric.
Yeah, so I'm going to stop sharing. Okay, so then it's, I think Mathieu is the second speaker.
Yes, I will take over. Thank you. Let me share my screen in a second. I hope it's visible now.
Yeah.
Yeah, thank you. So let me introduce myself. I'm Mathieu Jonin, working for Finovatis, consulting firm in innovation. I'm part of the Genial coordination team, which is led by Eric, and as such, I'm a co-leader of Work Package 5, which is mainly dedicated to the project management, intellectual property, data management, as well as ethical management. As Eric mentioned, we have a very high ambition, and we wish to be a game changer in ALD, HCC, both detection and prevention. So, of course, we had to gather this top-level expertise from 13 partners, which was well described just before. And for a pretty long period of time. If you want to succeed, of course, you have to take the appropriate management measures, and this is really our role here, making sure that we respect the contract with the European Commission. As Eric mentioned, we received quite a lot of money to fulfill a number of important objectives that will be described in details by my colleagues just afterwards. But basically, we have to make sure that we fulfill these objectives. In case there is any deviation from the work program, which you can consider as the contract, we have to define strategic and operational solutions that the partners can actually apply, and we have to make sure also that we respect both the ethical rules and the data privacy principles throughout Europe. Last but not least, we also have to make sure that we can track the expenses, because you saw that the budget is pretty high, 13 million euros from public money. So of course we have to demonstrate the European Commission that we use this money really accordingly to the plan and only for the benefit of the project. So in a nutshell, this is our, let's say, day-to-day activities for the 5 years, but also we have to take into account what is going to happen after the period of funding of Genial, and to make sure that our result and outputs will be sustainable in the future and we have a long-lasting impact. Mainly, we are not here only for science, but also to make sure that our outputs will be translated into practice for the benefit of patients. It will be described in detail afterwards, but really in a nutshell, we hope that we can create new tools for a better diagnosis, that we can stratify the patient, also identify population at risk. So this is clearly what we want to translate to application. And for this, our role as Work Package 5 is to make sure that we have a clear roadmap and that we can help our partners to fulfill this roadmap and make sure that the outputs from the scientific perspective will be translated for the benefit of patients. In this perspective, we have two major items. First, to make sure that this will be spread over Europe. So we have to make sure that we have the appropriate measures to protect the intellectual property related to this new knowledge, and make sure that it will be put into practice as soon as possible, not in 5 years from now, because there is a critical need, as was well described by Eric. Just one last word before I give the floor to my colleagues. I would just like to mention that we also coordinate a number of efforts in link with Cancer Mission Europe. I will not get into details because it's a quite complex organization, but let's say that the overall aim is to provide recommendations to shape the policy related to cancer in general, to help the European Commission for the coming years. And in this particular action, I would like to mention that ELPA has a key role in coordinating the effort within Genial and with other projects funded by the EU on how to address inequalities of access to quality care in Europe. And I would like to thank especially Veronica and Milan for their hard work in this perspective. And thank you for now, and I will be happy to take questions after my colleagues have finalized their presentation. Thank you.
Thank you very much, Eric. Thank you, Mathieu. Thank you for your kind words. We are looking forward to work on this report on inequalities. Thank you for the overall presentation of the aims for the project, and I'm giving floor to Dr. Patricia Patricia de la Cruz Ojeda. Welcome, and the floor is yours.
Thank you so much. I think that you all can see my presentation, right?
Yes.
Perfect. So as you have said, my name is Patricia. I'm working in France and I'm part of the work package 1. So this is what I'm going to present you today. And work package 1, the leader is INSERM. It's one of the most important institutions for research in France, in particular in clinical research. Our work package is led by Jessica Zuckman Rossi. We are working in the Centre de Recherche de Cordeliers. We are in Paris, but our work package is co-led by Professor Luca Valenti. Today we have with us 2 members of his team. We have Serena and Vittoria. But in this work package, we have some other participants from Brussels, mainly with the contribution of Eric Trabot, but also from other scientists from Europe and specifically from Germany with the participation of Stefan Buch. So as Eric has very well explained, ALD is a matter of concern. And it is responsible for most of the cirrhosis cases in Europe. Liver cancer is a very complex disease that usually sets up on chronic liver damage, on cirrhosis. And cirrhosis is very complex. It can be due to different etiologies. In particular, in our project, we focus on the chronic inflammation that is led by alcohol consumption. And in fact, even if it is a matter of concern, to show you some data, as Eric has very well done, many of the deaths, not only related to liver cancer, but in general terms are related to alcohol, and 36% of all liver diseases are related to alcohol abuse. And what is very important is that most of the deaths occur at working age, that impacts daily life and working countries. So even if this is a matter of concern, and it's very important, only 5% of the active clinical trials focus currently on ALD. So here comes Genial. So just to introduce a little bit the concept of genetics, our information that defines who and how we are comes in our genomes, comes in DNA, and it's a 4-letter code that defines how genes are expressed. So DNA can come in different versions. That is what we call in genetics a variant. That would be different variations in the DNA sequence that would be very similar to the different flavors of an ice cream. The base is the same, but at the end, there are different changes that make that there are different versions, and these different versions, in fact, are different consequences of gene expression. So these DNA variants, these different sequences, we can say that they could be germline variants, that those are inherited. Those are those variants that came from our parents and we can transmit to our children, but we are also subjected to different exposures during life. Those are those somatic variants that impact also gene expression, and they can be influenced by age, smoking, pollution, the daily habits, but also specifically in general by alcohol consumption. So both these germline variants and these somatic variants can impact the progression of a healthy liver to liver cancer. The contribution of somatic variants in liver cancer is well known, but there is a gap because the role of these germline, these inherited variants in liver cancer and specifically in alcohol-related liver cancer are poorly described. So Genial Work Package 1 focuses specifically in the identification of novel inherited genetic variants predisposing to alcoholic liver cancer. In particular, we can find two big different types of variants according to the frequency that we can find in the population. We could say that some of them are rare because we can find them in a very small proportion in the population, where others are common. That is, that they are most frequently found in the population. Let's say that rare variants can cause a major impact in gene expression. So to study these inherited germline variants, the work package 1 has different objectives. The first one is the creation of a case control dataset. That is, we need the patients to study these variants, and for that we have a lot of patients. The two main cohorts that were described in the proposal are the French-Belgian cohort and a German-Italian cohort of patients, but we are including a lot of samples in order to get a higher sample size and a high power in our analysis. Then we need to identify the common variants that will be led by the ULB, by Professor Eric Trepot. Then in objective 3, we will identify rare variants. That is the part that we're going to do in Paris. And then we need to validate our results in the general population. For that, we will use UK Biobank, but we have other sources of data and samples. And in non-alcoholic fatty liver disease, because fatty liver can happen not only in ALD, but also in non-alcoholic fatty liver disease, and both pathologies share some similarities. And this final objective will be led by the University of Milan. So for studying the common variants, at the end, we will use around 8,000 patients, including cases and controls. The technology that we will use is called GWAS. It's Genome Association Study. It's like taking a photo, a pixel photo, in which we can know what's going on. We can imagine what's inside, but we cannot go into specific details. For that, we used chips that contain predefined variants that we are going to study. The price, as you see, is very, very cheap, so we can have a lot of patients in the study. And with that, we want to extract the variants that are related to the condition, to the disease, to the liver cancer, and to evaluate involvement in the disease. And then to identify rare variants, we are going to do a more targeted approach. We are going to get the high resolution picture. And for that, we are going to read DNA. We are going to perform DNA sequencing. We want to know exactly the whole sequence that is in the DNA to get the whole picture of the variants. So for that, we are going to do whole genome sequencing in 350 patients to study the somatic variants. And then we will perform targeted sequencing to get the germline variants, because we think that the genes that have this somatic alteration will also have germline variants. However, the price of the technologies are lowering, so we might shift to broader sequencing, but we will see in some months. So with all these tasks, we will get hopefully a list of ALD risk genes harboring DNA damaging variants that will open the path to work package 1 and work package 3. So thank you very much for your attention.
Thank you very much, Dr. De La Cruz Ojeda. very nice presentation. And I'm opening the floor and giving the floor to our next speaker, Dr. Tina Tricot.
Hi, I will share my screen. Can you hear me?
Yes.
Okay. Can you see my slides?
Yes, we see the slides.
Oh, I'm in the wrong slide, sorry. So I'm Tina Tricot. I'm the project manager in the lab of Sarah Maria Vent, who is a part of the VIB, the Flemish Institute of Biotechnology in Belgium. And she is actually the lead of the work package 2. However, she could not be present, so she asked me to present today. And the co-lead is Mario Brosch, who is a PI at the Technical University of Dresden in Germany. So what is work package 2 about? It's about the evaluation of gene environment interactions for the risk of alcohol-related hepatocellular carcinoma development at molecular level. So as you may know, food has an influence on the liver environment. So whatever nutrition you take in has an influence on basically the lipids or the fats in your liver, the glucose in your liver, and other nutrients in your liver. So just to make it a little bit more easier in the next couple of slides. So when I'm talking about lipids in general in the liver, we talk about lipidome. While when we're looking at metabolome, which we talk about the other nutrients in the liver, meaning sugars, nucleotides, which are the building blocks for DNA, amino acids, which are the building blocks for proteins, and so on. So it is known that when we consume a lot of alcohol, that this alters the nutrient availability in liver. And it is known that actually the chronic alcohol leads to an increased lipid accumulation in the liver that gives rise to then a fatty liver and eventually cirrhosis and hepatocellular carcinoma. So moreover, it's also known that certain gene variants, as Patricia explained very nicely, are known to increase the lipid availability in the liver and thus enhance the risk for fatty liver and eventually hepatocellular carcinoma. So there is a real connection between genetic variations, liver environment, and also the increased risk of this hepatocellular carcinoma caused by chronic alcohol consumption. So therefore, we know that there is a big risk, but it's also important that we actually better understand that what these interactions are between the genes and the liver environment and the nutrients in the liver, to basically be able to see what happens early onwards in the development of alcohol-related hepatocellular carcinoma, and to find actually potential drug targets that we can then use to counteract this. So therefore, the work package 2 objective is actually to characterize how known and also novel inherited genetic variations, which will be identified in work package 1, interact with the nutrients in the liver, so that we get a better insight in this alcohol-related hepatocarcinogenesis and to identify potential targets. So how will we do this? We will basically look at 4 common and 1 rare genetic variants, and we will do some analysis on that. We will characterize histological differences. What do I mean with this? And we will see if there are differences in cell morphology within the natural tissue environment. Secondly, we will characterize metabolic and lipidomic differences. So we will look into the nutrient environment of the liver and see if lipids and metabolic or metabolites are differently expressed or present between the genetic variants. And then thirdly, we'll also characterize transcriptomic differences. What are transcriptomics? These are actually the differences in gene expressed in the cell. So basically these genetic variants can cause differences in genes that are expressed in the cells, and we will also try to characterize these. So how will we do this? So we will get from our partners in INSERM, Université Libre de Bruxelles, Assistance Publique Hôpitaux de Paris, and the Technische University of Dresden, we will get liver biopsies. And then we will slice this in very thin slides, which can then, we will then do analysis on consecutive slides, meaning that similar cells are present on these slides, which will help us with later onwards looking to all the different analyses on top of each other. So on these first slides, we will do histopathological analysis. What does this mean? We will use a stain and we can actually then see what the morphology of the cells are. And this will be done by the Assistance Publique Hôpitaux de Paris. On the third and on the second and the third slides, we will analyze metabolites and lipids. So we will look into the nutrients that are present in the cell, and we will use a technique called mass spectrometry imaging for this, which is basically a laser that will shoot at a 50 micron resolution. So that means a 1/20th of a millimeter. It will shoot a laser, and then metabolites will be detected for each of these 50 microns. So this way we can, in the entire tissue, see which nutrients are present in the cell, almost at single cell level. And this will be done at the Flemish Institute of Biotechnology in Belgium. And then on the fourth slide, we will do spatial transcriptomic analysis. This means that we will look at which genes are expressed. So also at the 50 micron resolution, we can then see for each of these spots basically which genes are expressed. And we will, and thus present in this, and thus determine the fate of the cell. So we will enrich this with also single RNA sequencing. What this is actually a technique where we, for each cell, also find the full list of genes that are expressed in the cell, and this is to basically enrich our spatial transcriptomics. This is done by the Technische Universität of Dresden. So we will have then basically the information of the cell morphology, the metabolites present, so the nutrients present, the lipids present in the cells, as well as the genes that are expressed, and we will overlay all this data, and this will be done by the University of Paris-Cité, and this will help us via data integration to actually identify some hypothetical mechanisms and gene environment interactions for the investigated genetic variants. So, of course, we then have to validate this hypothesis. So the idea would then be to generate genetic mouse models with the genetic variants of interest. So basically the ones that were also identified in work package 1, and validate if this, first of all, these mouse models can recapitulate the disease found in the patient. And moreover, we will also then, of course, validate the hypothesis that we generate from all the data that we generated. And we will basically, this multi-omics integration will give us some ideas of which processes in the cells might be changed between the genetic variants, and thus might be interesting as drug targets. So we will select some potential molecules that might attenuate the hepatocellular carcinoma and test this also in the preclinical models. And this will be done by Champalimaud Foundation. So what do we expect is that this information will advance our understanding of the alcohol-related hepatocellular carcinoma onset, its evolution, and provide also some potential chemopreventive drug targets. So thank you for your attention. And if you have questions, you can ask probably after everyone has spoken.
Thank you very much, Dr. Tricot. Very clear presentation on such a complex topic. Thank you very much. And I'm giving the floor to our last speaker today, Professor Nahon. The floor is yours.
Hi, can you see my screen? I'm trying to...
Not yet. Ah, yeah. Okay. Yes, we can see them, the slides.
Okay. Thank you. Hi, everyone. So we have seen some very nice presentation of very basic elements of the project. Now we'll switch to the more clinical aspects, which is basically work package 3 of the Genial project. This is a slide that tries to maybe complexify a little bit what has been said by Eric at the beginning, where you have this idea that patients drink alcohol, they get cirrhosis and they get cancer. But it's in clinical practice, it's more heterogeneous because patients, of course, they drink alcohol, but they also may have some features of the metabolic syndrome. They may have hepatitis B or C that may have been cured or controlled by antivirals. So it's a very long story for many years and decades until the patient develops cirrhosis. And ultimately, they may develop liver cancer, but also other competitive events, such as complication related to portal hypertension, liver failure, also complications linked to comorbidities such as cardiovascular disease, extrahepatic cancers. Anyhow, it's a very complex, long story. And if we want to applicate what we'll be finding in work package 1 and 2 into this complexity, we will need some real-life cohorts of patients with a long follow-up to really understand what is going on in these patients. And particularly, this is the case for hepatocellular carcinoma early detection. This is something that I know that patients are looking forward, because cirrhotic patients are the ideal target for HCC screening. And the whole idea of screening liver cancer is to find small liver cancers. You see here on the left panels where patients are eligible for curative treatment. And this is an example of a patient that have a central hepatocellular carcinoma here that can be quite easily treated in a curative way. For example, here, this is an example of a percutaneous ablation. And by means of this curative treatment, you can put the patient in remission and expect a survival above 5 years. And this is in sharp contrast with what we can call this advanced setting of liver cancer. And usually these patients were not diagnosed with cirrhosis and were not included in surveillance programs, because in these patients, as you can see, the tumor burden is too heavy to implement curative procedures. So patients are treated by palliative treatment, and the life expectancy is quite low, below 2 years. But unfortunately, in our centers, tertiary centers, most patients have advanced HCC. So this is a strong need, and I know that ELPA is very aware of that in its communication with patients on how we could change this balance and have more patients with small HCC. So to do so, we perform liver ultrasound every 6 months in patients with cirrhosis. This is a standard of care, but we know in clinical practice that it's not really a performance tool, because if we focus on the main endpoint, which is detecting very early tumors below 2 centimeters, we know that the sensitivity of ultrasound is below 30%. So this is why we've been developing some new devices, some new tools, such as imaging techniques or circulating biomarkers. But of course, we cannot implement these expensive tools in all comers, because it is very expensive. And for example, MRI is not only expensive, it is also not available everywhere for everyone, because we know that there is a queue list to get an MRI for diagnosis purposes. So we have to choose which patients should undergo these very expensive and complicated detection tools. So this is basically the idea of the personalization of screening, which is summarized here on this slide, where we would have some patients with cirrhosis that are considered at low risk of developing liver cancer that should only undergo ultrasound surveillance every 6 months. Patients with intermediate risk in whom we could reinforce these programs using education programs, mail outreach, dedicated clinical pathway, etc. And patients with high risk of liver cancer in whom we could optimize surveillance using more expensive tools because of this increasing cost of surveillance that could be only dedicated to the high-risk patients. So this is, I would say, a personalization and a more accurate use of these limited resources in clinical practice. And this is exactly what we're trying to do with Work Package 3. We're trying to really understand how research from work package 1 and 2 could be implemented in these algorithms that aim at defining who is at risk, high risk, low risk, medium risk of developing liver cancer. And for this, we plan to use patients from Belgium, France, and Italy, included in prospective cohorts undergoing surveillance for liver cancer. And we will try to both study the role of genetic variants, but also the environment, which is basically the main theme of the Genial project, and combine with also imaging data from biopsies, from imaging techniques. And we will try to create some new algorithm using solution based on artificial intelligence to really define who is really at risk and who should be included in reinforced programs. So to this aim, I come back to this slide. We will use nearly 3,000 patients who have a prospective follow-up during surveillance programs. And we know that these patients that are already in prospective cohorts, we have some constitutional DNA for most of them. We have some pathology slides available, and also some images from CT scan and MRI, and also a liquid biopsy from a plasma that have been drawn every year. So we'll use all these samples to generate these new algorithms. And we will also validate these findings in an ongoing clinical trial, which is exactly what we would like to see in the forthcoming years. This is a trial called Fast Track, and you see that it proposes to randomize patients with a very high risk that are selected using a very simple scoring system, a clinical scoring system, between those who will have only ultrasound and those who will have only ultrasound and MRI for liver detection. And for this patient that are currently under enrollment, we have constitutional DNA. Because it is prospective, we have all results from surveys regarding environmental factors, and also results from imaging techniques. So my last slide is, maybe some of you were there 2 years ago. We had this meeting with the European Parliament in the setting of the EU Beating Plan Cancer. And at this time, we were asked to highlight how improving HCC screening in European patients is a very critical issue for our patients. And you remember that we discussed that, of course, in the primary care setting, identifying patients with exposition to chronic liver disease and those with cirrhosis is crucial in order to put them in the clinical ward and include them in HCC surveillance programs. And clearly, you see that the Genial project is basically aimed at improving HCC risk stratification and refinement of surveillance programs, which all need dedicated efforts from educational, compliance, clinical pathways, and research. And I think that with the help of ELPA and European Union, we will have some very nice conclusions to be held ahead in the forthcoming years. Thank you.
Thank you very much, Professor Nahon. Thank you to all the speakers. So I'm opening the floor for any questions, comments from the audience. So we have patient representatives, caregivers, and also medical experts. So please let us know if you have any question, comment. Okay, and I see, so I see a comment from Dr. Glasova from Slovakia.
Good evening. Congratulations to your huge and wonderful project. My first question is about what sort of environmental pollutants are you going to look into? And the second would be, what type of MRI imaging methods are you going to use within the project? There was artificial intelligence mentioned, so just these simple questions. Thank you.
All for Pierre, I think.
Yes, thank you. I didn't hear well your first question, so but I can
I can repeat. I was listening carefully, and I heard that there will be also environmental pollutants studied. So I would like to know, because I'm curious, how and which. What are you going to look into? Thank you.
Okay, so basically your questions are directed to the ongoing trial for MRI versus ultrasound. So because it is, of course, really prospective and ongoing, we have the opportunity to implement questionnaires about environment, which is a very, very important part of the Genial project, because you have seen that on the one part there is the genetic, which can be studied on samples, but the environment, of course, can only be prospectively assessed, because if you try from a medical files to understand how many cigarettes the patient has have been smoking, or alcohol, it's very biased. So we have to address specific questionnaires on alcohol consumption, cigarettes, food, socioeconomic determinants, and also the whole environment of the patient. So we have a dedicated team in University Paris 13, in my university. You know, they are the one who created the Nutri-Score. You know Nutri-Score? It's a scoring that we have, you know, in the supermarket. About it gives you a very simple A, B, C, D, E. So any kid, my kids can understand what it means. When they buy cookies, they say, no, not this one, because it's Nutri-Score D or E, it's not bad for your health. So they are very, very keen on addressing questionnaires to patients, to general population, on what is the global environment on all status. So it's not really directed to pollution, for example, but there are some questions that will be focused on where do you live, in which part, is it in the countryside, in the city, in the suburban area, which floor, and so you have a lot of items in the questionnaires. It will be one-shot questionnaires, but we will be able to correlate it with the whole clinical history of this patient that will be followed up for many years. So, and of course, we'll be able to correlate with genetic findings we will apply in this cohort. So it will be very, very interesting, I think. And regarding MRI, it will be abbreviated MRI, which is, you know, because when you perform an MRI, total MRI, you inject some gadolinium and everything. It's long, it takes time, 30 minutes. Patients are claustrophobic, and it's very expensive and not available for everyone. So we have to make compromises. And clearly for liver cancer, abbreviated MRI have been shown to be very effective. It has a very high sensitivity, above 80% as compared to 30% for ultrasound. So by 10 minutes, using some dedicated sequences without injecting contrast, 10 minutes, you can have a very good performance for detecting very small liver nodules that can be liver cancer. So, and it's also the case maybe for breast cancer in the forthcoming years in other trials. So MRI and abbreviated MRI is a hopeful tool for early detection in cancer.
Okay, thank you. Thank you very much.
Thank you, Professor Nahon. Moving forward, there is another comment from a patient representative from North Macedonia. Milan, the floor is yours.
Thank you very much. First of all, that we have the pleasure to listen to you. Liver cancer is among the highest priority of ELPA and its members. It's familiar that we trying to push the early detection on the high level political agenda to elevate the liver cancer screening in the next period. And I saw a lot of interesting, let's say, some kind of mentioning of different specific genetic biomarkers, markers for individuals. What are you planning exactly to do in that field? I think was in the first slide also new biomarkers was mentioned from Matthew. I think I saw the beginning, and because this is very hopeful for the liver community, if we have some signs that can improve the early detection maybe in the future with a new biomarker which give a bigger sensitivity, can we establish maybe a program completely together with the European Commission about the liver screening in the cancer?
Lucia, you want to answer that, or you want me to answer?
I mean, I can start, and maybe you can complement the answer if you want. Yes, is of course the idea is to get these variants that predispose to liver cancer, and of course the idea is not to get this data to go to a drawer. The idea is, of course, to push and to try to develop biomarkers of prevention and early detection. The work package 1 aims to identify these variants because there should be, but we need to investigate a little bit more. I would say that in work package 3, we have some tools of circulating tumor DNA that complement this approach of biomarker and establishing diagnostic tools. In particular, in work package 1, the aim is not to establish a diagnostic biomarker, let's say, but in the future, of course.
I think we really have to be careful when we talk about genetics in complex diseases. So complex disease means anything, could mean any diseases. Also, when you talk about trace, like your weight, your height, we should not forget that what is the main driver of the disease is environment. So it is clearly alcohol that is the main driver. And it's important, of course, to identify genetic variants. I'm someone who has been studying genetics, and this is my main area of research. It's extremely useful to understand what is happening in disease. But when it comes to, let's say, screening and how it's going to impact the outcome, I think we should be extremely careful here. Because first, it's misleading. What we should work and aim for is make sure that individuals, patients, try to drink as low as possible. As a lower amount of alcohol as possible, because this is really the main driver of the disease. And I'm not aware of a single disease where genetics can really, predict the outcome and predict the individual risk of an individual. I think it contributes to, as I said, to understand, to find maybe targets, but there is no way that in a disease that is common, frequent, that genetics is going to play a high role. Because just for natural selection, if you have something where genetics impacts extremely, has a very strong impact on the phenotype, on the outcome, then it's going to be extremely rare by definition, because that is nature. When something has a huge impact, it doesn't survive. So genetics plays a role, but this is really the thing with Genial. Let's not forget the environment, and especially in the context of this meeting here, what is extremely important, we should not mislead the patient and individuals. What is important is that the level of alcohol that is consumed is as low as possible.
I fully agree with Eric, because especially with genetics, when you talk about genetics to patients, to the common European population, they have this idea that genetics is going to give all the answers. And if you have the good genes, well, you can drink or do whatever. You have the bad genes, it's done for you. So but it's more complicated than that. And this is why this is the call of the European mission. It was environment and gene. Understanding how it works. This is work package 1 and 2. And then in work package 3, I highlighted how it is complex in clinical practice, and there is no alcohol, hepatitis viral here. The patient usually have different influences of their environment and their genetics. In the end of the day, we don't know what we are going to find, but again, as Eric has stressed out, it's a combination of both environment and genetics that may help to improve the management of patients. But understanding is also, of course, very important.
If I have the time, I would say that the summary is that genetics is here a complementary approach. In fact, Genial is that. In work package 1, we have genetics, molecular data in work package 2, and clinical data in work package 3, and that is the good combination, let's say.
for me, genetics is even more useful to find, for example, a drug. When you have when you, for example, find a gene that plays a role, then maybe you can find a drug. And this is for me 10 times more important than predicting how someone is going to, what is going to be the outcome of a given individual, because this is not true. We do not, not everything is written in gene, everything is written in fact in your environment, and your genetic background modulates it, but that's not the main driver.
Thank you very much for amazing presentations. Thank you also for answering all the questions from the patient community. For our audience, we will be back with the second Layman event in a few years, when the project will be at its last stage, where we will be able to share with you the results of the project and also share with you the steps in the future because of the results the project performed. So stay tuned, follow us on social media, and until then, goodbye.